Hi, good morning, friends. This is Mr. P.R. Thakare, Assistant Professor, Department of Zoology from Jizamata Mahavidyalaya, Buldhana. Today, through this online lecture, we are going to discuss on topic antigens and its origin, types, and what is the hapten molecule. So we will see everything in details about the antigen, its types, origins, and hapten molecules. So let's start with this video. Okay, so starts with the first terminology, that is humeral immunity. That humeral immunity only results in the production of proteinous molecules, and that proteinous molecules which is released by the humeral immunity results that are known as the immunoglobulins or the gamma globulins, or sometimes it is also known as the antibodies. So at the right side you can see Y-shaped antibodies in the bluish color you can see there. So the humeral immunity only results in the production of immunoglobulins or the antibodies or gamma globulins. So our body exposed to sometime foreign material termed as antigens, which may be harmful to our body. That antigen, it may be in the form of viruses, bacteria, protozoans, helminths, and the other cellular bodies, etc., in the form of antigen and exposed to our body. That antigen has bypassed other protective mechanism, that is first and second line of defense. Based on immunogenicity, that antigen can be categorized into two types. One is complete antigen and another one is the incomplete antigen. So complete antigen, that are those substances which can induces or enhances the antibody formation by themselves and can react specifically with these antibodies. सो कंप्लीट एंटीजेंस ऐसे सब्सटेंसेस रहते हैं जो एंटीबॉडी प्रोडक्शन के लिए रिस्पॉन्सिबल रहते हैं एंड दे कैन ईजिली specifically reacts with the antibody molecules. So these are called as the complete antigen molecules. Second type on the basis of immunogenicity can be in front of us, incomplete antigen. Sometimes it is also known as Haptens molecule. That incomplete antigens or the hapten substances cannot induces the antibody formation on its own, but can become immunogenic when covalently linked to proteins called as the carrier proteins. ये जो इनकम्प्लीट एंटीजन मॉलिक्यूल रहते हैं वो एंटीबॉडी प्रोड्यूस नहीं करते हैं, लेकिन वो इम्यूनोजेनिक रिस्पॉन्स कैरियर प्रोटीन के साथ में लिंक होने जाने के बाद में वो एंटीबॉडी प्रोडक्शन कर सकते हैं. सो इनकम्प्लीट एंटीजन आर ऑफ हैविंग टू टाइप्स. वन इज़ कॉम्प्लेक्स एंड अनदर इज़ द सिंपल. So complex incomplete antigen and simple incomplete antigen. Now based on origin, the types of antigens can be categorized into following types: exogenous antigens and endogenous antigens. Exogenous antigens are antigens that have entered in the body from the outside. For example, by inhalation, ingestion or injection, the immune system responses to exogenous antigen is often subclinical. इट वॉज अबाउट द एक्सोजिनस एंटीजेंस, जो बाहर से एंटीजन हमारे बॉडी के ऊपर आते हैं, अटैक करते हैं थ्रू इनहेलेशन, इंजेक्शन और इंजेक्शन, उससे कहते हैं एक्सोजिनस एंटीजन. नेक्स्ट टाइप इज एंडोजिनस एंटीजन. दैट एंडोजिनस एंटीजेंस आर एंटीजेंस दैट हैव बीन जनरेटेड विद इन प्रीवियसली नॉर्मल सेल्स एज अ रिजल्ट ऑफ अ नॉर्मल सेल मेटाबॉलिज्म और बिकॉज ऑफ वायरल और इंट्रासेलुलर बैक्टीरियल इन्फेक्शन. ये जो एंडोजिनस एंटीजेंस है वो हमारी बॉडी के अंदर मेटाबॉलिज्म के समय में और जब इंट्रासेलुलर बैक्टीरियल जो इन्फेक्शन होता है तब एंडोजेनस एंटीजेंस बॉडी के अंदर सीक्रेट होते हैं, सीक्रेट किए जाते हैं नॉर्मल सेल्स के तरह से, तो उसे कहते हैं एंडोजेनस एंटीजन. सो दैट टू टाइप्स आर बेस्ड ऑन दी ओरिजिन, एक्सोजेनस एंटीजन एंड एंडोजेनस एंटीजन्स. नाउ फैक्टर्स ऑफ एंटीजेनिसिटी, which molecules we called it as the antigens. Certain factors should be followed by that particular type of molecule, then we call that molecule as antigen. So factors of antigenicity, one is foreignness. That antigen must be foreignness to immune system. What substances are foreignness to immune system? It's a question in front of us. According to the Burnett's clone selection theory, foreignness means the non-self means substances that never contact with lymphocytes during embryo period. फॉरेननेस एक प्रॉपर्टी है, एक फैक्टर है. ये फैक्टर अगर मॉलिक्यूल बियर करता है, पजेस करता है, तो उसे हम एंटीजन कह सकते हैं. दैट मॉलिक्यूल never contact with the lymphocytes during embryo period, and we called that molecule as a antigen. Size: the larger molecules are highly antigenic. If the substances has large size, that large molecule are highly antigenic. Lower molecular weight are either non-antigenic or weakly antigenic. जिस substances की size या फिर molecular weight कम होता है, we called it as the non-antigenic or weakly antigenic. Second factor is size. Now third factor is chemical nature. Most naturally occurring antigens are either proteins or the polysaccharides. So antigens must be or that particular substance be proteins or the polysaccharides. Then lipids and nucleic acids are less antigenic on their own, but do so when combined with proteins. The substances, if it is lipid or nucleic acid, we don't call it as the antigen, because lipids and nucleic acid are generally less antigenic. But if that lipid and nucleic acids when connected or linked to the proteins, then that particular substance will be act as the antigen, and we called it as the antigen, exogenous antigen or the endogenous antigen. Fourth factor is susceptibility to tissue enzymes. Substances which can be metabolized and are able to the action of tissue enzyme behaves as antigen. एक factor important है. Fifth factor is antigenic specificity. That substance specifically binds to the specific antibody, means specificity is must there. That molecule depends upon epitope. Position of epitope in the antigen molecule is important for the specificity. So the epitope site should be recognized by that particular substance, then we called it as the antigen. A species specificity, it is also one of the important factor of antigenicity. Tissues of all individual in species possesses the species specific antigens. Isospecificity, it depends on the isoantigens which may be found in some but not all members of species. Autospecificity, self antigens are generally non-antigenic, but in some cases such as Lens protein and sperms, these are not recognized as the self antigen because they are absent during the embryonic life and they develop later. See that Lens protein means eye proteins, and the sperms, that is germ cells, these are not fully formed during the embryonic life. They are developed after the embryonic period is completed. So the Lens protein and sperms, these are not recognized our self antigens, and they can easily specifically binds to our self antigens or the antibodies. Hence that specificity should be recognized there. Organospecificity, it is also one of the important factor of antigenicity. Some organs such as the brain, kidney and Lens protein of different species share the same antigens. Such antigens are the characteristics of an organ or a tissue found in different species, and they are known as organ specific antigen. Heterogenic specificity, that particular substance or the molecule also possesses the heterogenic specificity, then we call that substance or molecule as antigen. The same or closely related antigen may sometime occur in different biological species, classes and kingdom are now as heterophile antigens. Antibodies to this antigen produced by one species can cross-react with the antigen of other species. For example, Postman antigen, Will-Field RXN antigen and the Paul-Bunel test. That the three things heterogenic specificity shows. Then nature of immunogen, foreignness. In order to elicit an immune response, a molecule must be recognized as the non-self by that host. What kinds of substances can be foreignness to immune system? Heterogeneous substances, various pathogens, xenoantigenic tissues, then allogenic substances, grafted allogenic tissues or organs, antigenic components that never contact with the lymphocytes during the embryo period. Molecular size also, as we have seen in previous slide. Haptens, let's turn toward on the next point, that is haptens or the incomplete antigen. The term hapten was given by the immunologist Karl Landsteiner, who studied them in early 20th century. That haptens was discovered by the Karl Landsteiner in the early 20th century. That haptens came from a Greek word hapten meaning to fasten. That haptens is nothing but has a meaning to fasten something. Haptens are low molecular weight molecules. It is one important character of the hapten molecule. Second, small organic molecule that are antigenic but not immunogenic. If any small organic molecule in front of us, then we called it as are antigenic but not immunogenic. That molecule are nothing but the hapten molecule. Haptens that they can bind to immune cells but fail to induce the humoral or cell mediated immune response. That hapten molecules when binds to any immune cells, for example B cell, T cells, or any monocytes or neutrophils immune cells, then that hapten molecule does not induce the humoral or cell mediated immune responses. That hapten, hence no antibodies are raised against them. That hapten molecule does not raise any antibody against it. Haptens can induce immune responses when covalently bound to a large protein called as the carrier protein, forming immunogenic hapten carrier conjugate. ये hapten molecule तभी नॉर्मल सेल्स को इंड्यूस कर सकता है, एंटीबॉडी को प्रोड्यूस कर सकता है जब वो कोवेलेंटली बाउंड हो जाए कैरियर प्रोटीन के साथ और इम्यूनोजेनिक हैप्टेन कैरियर कॉन्जुगेट बनाए. जैसे ही वो इम्यूनोजेनिक हैप्टेन कैरियर कॉन्जुगेट बनाता है वैसे ही जो इम्यून सेल्स है इन द फॉर्म ऑफ बी सेल्स, टी सेल्स और दी मेजर हिस्टोकम्पैटिबिलिटी कॉम्प्लेक्सेस सेल्स जो है वो एंटीबॉडीज प्रोड्यूस कर सकते हैं, इंटरल्यूकिन इंटरफेरॉन प्रोड्यूस कर सकते हैं. सो हैप्टेन्स मस्ट बी बाइंड विद द covalently to large protein molecule called as carrier protein molecule, forming the immunogenic hapten carrier conjugate. The carrier protein molecule are antigenic as well as immunogenic. See that carrier protein is going to be bound with the hapten molecule. That carrier protein molecule must be antigenic as well as immunogenic. You can see here hapten molecule when bind to the carrier protein molecule, that carrier protein molecule induce the B cell stimulation for the formation of antibodies there. See that hapten molecules itself does not activate the B cells, but that hapten molecule when bind to the carrier protein molecule, then that hapten carrier protein conjugate is produced there, and then that complex or the conjugate of hapten and carrier protein molecule easily activates the B cells. See here hapten, it is showing in the pinkish color. It binds to the antibody Y-shaped. It is in the form of BSA, and antibody to hapten easily junctions of hapten and BSA easily activates the B cell activations. Animals immunized with such a hapten conjugate produces antibodies specific for the hapten determinant, unaltered the epitopes and the carrier protein, and new epitopes formed by the combined parts of both the hapten and carrier molecules. So the hapten molecule only active when it binds to the carrier protein molecule. Thus hapten when combined with the carrier molecule, it will form the immunogen, and by itself hapten cannot function as an immunogenic epitope. हैप्टेन जो मॉलिक्यूल है वो अकेला खुद बी सेल एक्टिवेशन नहीं कर सकता है, वो इम्यूनोजन करके काम नहीं कर सकता है. दैट हैप्टेन मॉलिक्यूल मस्ट बी बाइंड विद द कैरियर प्रोटीन मॉलिक्यूल एंड दे विल फॉर्म देयर इम्यूनोजेनिक कॉम्प्लेक्स, and then that immunogenic complex will ultimately activates the immune cells. But when multiple molecules of a single hapten are coupled to a carrier protein or a non-immunogenic homopolymer, the hapten becomes accessible to the immune system and can function as an immunogen. There are few examples of haptens which are showing through this slide. Some examples is given here. Dinitrophenol is also act as the hapten molecules. Aminobenzene, O-aminobenzoic acid, M-aminobenzoic acid and the P-aminobenzoic acid. These are good examples of the hapten molecule. Many biologically important substances which also can function as haptens. For example, drugs such as penicillin, peptide hormones and certain steroid hormones also biologically important active hapten molecules which can initiate the activation in immune cells. Adjuvants is another terminology is here in front of us. That adjuvants are substances that when mixed with an antigen and injected with it, that increases or enhance the immunogenicity of that antigen. Adjuvants ऐसे मॉलिक्यूल होते हैं वो अगर एंटीजन के साथ में मिक्स हो जाए तो वो इम्यूनोजेनिसिटी नॉर्मल इम्यून सेल्स में बढ़ा सकते हैं. सो एडजुवेंट्स आर दोज सब्सटेंसेस व्हेन वी मिक्स विद द एंटीजन और इंजेक्टेड विथ it enhances the immunogenicity of that antigen. The word adjuvants were derived from the Latin word adjuvare, which means to help. Adjuvant का मतलब ये होता है कि किसी को help करना. Adjuvants are often used to boost the immune response when an antigen has low immunogenicity or when only small amounts of an antigen are available. एंटीजन की इम्यूनोजेनिसिटी बढ़ाने के लिए, और कोई एंटीजन अगर स्मॉल अमाउंट में है उसकी इम्यूनोजेनिसिटी और एंटीजेनिसिटी बढ़ाने के लिए एडजुवेंट मॉलिक्यूल हेल्प करते हैं. एडजुवेंट्स में बी एडेड टू अ वैक्सीन टू मॉडिफाई द इम्यून रिस्पॉन्सेस. एडजुवेंट वैक्सीन में भी मिक्स किए जा सकते हैं, एडिड किए जा सकते हैं इम्यून रिस्पॉन्स बढ़ाने के लिए, मॉडिफाई करने के लिए. The antibody response of mice to immunization with bovine serum albumin can be increased 5-fold or more if the BSA is administered with an adjuvant. जो बोविन सीरम अल्ब्यूमिन है वो उसका इम्यूनाइजेशन बढ़ाने के लिए एडजुवेंट मॉलिक्यूल हेल्प करते हैं उनको ऐड करने के बाद में. रूट ऑफ एडमिनिस्ट्रेशन ऑफ एडजुवेंट. अब एडजुवेंट कैसे, किस तरीके से, कौन से वो रूट से कि उसके माध्यम से एंटीजन के साथ मिलाए जाते हैं, फिर वैक्सीन के साथ में मिलाए जाते हैं. जनरली द सबक्यूटेनियस रूट इज़ द बेटर दैन द इंट्रावेनस और इंट्रागैस्ट्रिक रूट्स. सी दैट सबक्यूटेनियस रूट इज़ द बेस्ट रूट और द वे थ्रू विच एडजुवेंट कैन ईजिली एडमिनिस्टर्ड और इनकॉर्पोरेटेड inside the body cells. So subcutaneous route is the very important route for the adjuvant incorporation. The route of antigen administration can also alter the nature of the response. Antigen administered intravenously is carried first to the spleen, whereas antigen administered subcutaneously moves first to local lymph nodes. So intravenously rather than subcutaneously, effect shows by the adjuvant molecule. Adjuvant is an immunological agent which enhances the body's immune response to an antigen. That adjuvants may be added to a vaccine to boost the immune response to produce more antibodies and longer lasting immunity, thus minimizing the dose of antigen needed to the vaccine. That adjuvants are used in combination with a specific antigen that produced a more robust immune response than the antigen can do alone. Antigen खुद itself जितनी इम्यूनोजेनिसिटी नहीं बढ़ा सकता है, वह इतनी उससे भी ज़्यादा एडजुवेंट मॉलिक्यूल उसमें मिक्स होने के बाद में इम्यून रिस्पॉन्स को बूस्टअप करता है. उसकी स्पेसिफिक एंटीजन जो है वो स्पेसिफिक एंटीबॉडी के साथ में रिएक्ट होता है और इम्यूनोजेनिसिटी बढ़ाता है. सो एडजुवेंट हेल्प टू द एंटीजन. Adjuvant, many diverse classes of compounds have been assessed as adjuvant, including mineral salts also acts as the adjuvant, microbial products, emulsions, certain cytokines and the liposomes can useful as a adjuvant. Some of the common adjuvants that are being used in the vaccine that are alum, MF59, ASO3, ASO4, CpG, ODN, virosomes, etc. These are good examples of adjuvant which is used in the vaccines to fasten the rate of vaccines. Thank you. I hope that it is hopeful to you.